Good afternoon, everyone. Let's start the first session for today. My name is Kenta Nakai, and I will chair this session. So it is my great pleasure to invite Professor Steve Rozen as a keynote speaker. Dr. Rozen is the founder and the director of the Duke-NUS Center for Computational Biology, where they have published more than 250 scientific papers since 2013. Dr. Rozen's lab focuses on bioinformatics and cancer genomics. He created and maintains the widely used primer 3 software for PCR primer design. Perhaps you have used the software. Recently, he co-led a major study that used machine learning to computationally mine mutation data from almost 24,000 human cancer samples, and the work was published in Nature. So today, his talk is entitled Mutational Signatures. What causes the mutations in these cancers? Why do we care? Dr. Rozen, please.
Dr. Nakai, thank you very much. So it's my great pleasure to be able to present here this afternoon. And I think with that, I'm just going to share my screen and start. Okay, so again, I'm going to be talking about mutational signatures. And I'll start by describing why they're important. Let's see here. Okay, so first I'll talk about what are mutational signatures, and I'll talk about what they're good for. And then I'll talk about the state of the art of computational analysis of mutational signatures. And then I'll talk about some important unsolved problems in mutational signature analysis. And then I'll summarize. So what are mutational signatures and what are they good for? So we're going to be using the example of Aristolochic acids and their relatives, which we're going to just collectively call AA. It's a family of chemicals that are related. We're going to call them all AA. Plants with AA are widely used as herbal medicine. The AA occurs naturally in the plants. So here's some examples. These are all from Chinese medicine, but it's not, I should say that these are all plants in the genera. Hold on one sec. I can't find my mute button. Excuse me. Anyway, okay, these are all plants in Aristolochia or Asram groups. They're not just found in herbal medicine, so they're used in herbal medicine in Europe, in South America, and in India. They're well-known kidney toxins, well-known nephrotoxins. And AAs emerged as a human health problem in the 1990s when about 100 young women with end-stage kidney disease, all attending a single weight loss clinic in Europe, at which weight loss clinic they replaced one kind of fangji, Han fangji, with a different kind of fangji, Guang fangji. So this is the harmless one, Han fangji. It doesn't have AA. And this is the one that contains AA, Guang fangji. It's a totally different type of plant, but the dried roots look very similar. And it turned out that many of the kidney failure victims went on to develop upper tract urothelial cancer. So urothelial cancer is in the ureter, which is the tube that drains the kidney. So we're going to do now a quick review of somatic mutations. So every time our cells divide, mutations arise, and mutations that were not present in the fertilized egg are called somatic mutations. And our focus of mutational signature analysis is going to be on the somatic mutations. So we detect somatic mutations by DNA sequencing normal cells, and in this case, mostly tumor cells, not always tumor cells. And we look for the variants that are found only in the tumor. So here, there's a T here in the tumor, and an A here in the normal. And so we had a mutation from a somatic mutation from A to T. And we're going to mostly think about these in the context of the preceding and following nucleotides. So we're going to think of this as a CAA to CTA mutation. Okay, so I'm going to emphasize that we look at all the mutations. Most of the mutations are not causing the cancer. They're innocent bystanders. So our focus is on finding out what mutated the DNA, not what the mutated DNA did to the cell's behavior. So here's the example of the mutational signature of AA in an AA-exposed upper tract urothelial carcinoma. So each bar represents the total number of single nucleotide substitutions in a particular trinucleotide context, in the context of the preceding and following bases. So for example, the highest bar here represents the number of CAG to CTG mutations across the entire genome. And the higher the bar, the more CAG to CTG mutations. And then the next star bar here is AG to ATG mutations. There are quite a few, but not as many. And then there are very few mutations from C to A, C to G, C to T, almost none of these. So 6 years ago, we knew about the AA mutational signature in urothelial cancer, upper tract urothelial cancer, and in cell lines. As recently as 3 years ago, the mutational signatures had showed AA in multiple tumor types. But Taiwan is a known hotspot for AA exposure, but AA and liver cancer in Taiwan had not been studied. So we decided to study AA and liver cancer in Taiwan. So we sequenced 98 exomes and they matched normals, 98 exomes from liver cancer and matched normals. And quite to our surprise, 78% of the liver cancers had the AA signature. So going in, we had probably thought maybe 20% might be what we would expect. So we're quite surprised. It turns out that liver cancers actually have been pretty widely sequenced. So we were able to ask the question of how extensive is AA exposure in 1,600 liver cancers around the world. And we found that, again, maybe to our surprise, it was quite prevalent. So we found a lot of it, a lot of mutations in China, Southeast Asia. Taiwan remained the hotspot. There were some in Korea, very few in Japan, few in Europe. One hospital in the US had a lot, and we think this is because people came to that clinic for treatment from Asia. Just an example of the primary data that we looked at. So here you can see the gray peaks here that represent the AA signature. So I would assert that AA can cause liver cancer. The evidence is that it's a known mutagen and urinary tract carcinogen. We saw multiple known cancer driver genes with AA mutations in liver cancer. The proportion of AA-exposed liver cancers in Taiwan is much greater than the proportion of the population estimated to be exposed. AA addicts have been found in the liver tissue of liver cancer patients. There's a linear relationship between AA dose and risk of liver cancer in Hep B and hepatitis C infected patients. And AA causes liver cancer in mice. I think the evidence is quite strong. So that's an example of how mutational signature analysis can illuminate cancer epidemiology, and ideally this will lead to people avoiding consuming these herbs and also maybe some regulatory changes that would make it harder for people to be exposed to these herbs. So now I'm going to switch gears to the computational analysis of mutational signatures and sort of what we know today. So I'm going to talk about the questions we want to answer. So one question we call extraction or discovery. So this means that given a large number of spectra, that is mutational spectra like the ones we just looked at, what mutational signatures are present. So from a machine learning point of view, the mutational signatures are latent variables to be discovered. And we'd also, at that time, want to know how many mutations are caused by each extracted signature in each spectrum. And in the jargon, we're calling that attribution. Then there's another problem, which is we call attribution only. So if you're given the known set of mutational signatures and one or more spectra, you want to answer the question, how many mutations in each spectrum were caused by each signature? And then finally, something that was motivated by our work on the Aristolochic acid and liver cancer, given known mutational signatures, what is the evidence that a signature of interest is present in a spectrum? So in our case, we had the spectra of these liver cancers, and we wanted to know, answer the question as rigorously as possible, what's the evidence that the AA signature is present in that spectrum? So we'll focus first about extraction or discovery. So many signatures have been extracted, and many of these have been confirmed by additional evidence, either by experimental evidence or by correlation with other factors. And sometimes the spectrum is dominated by one signature. So these are actually the spectra of these cancers, but they're dominated by the signature of AA. So when you look at them, you basically just see the signature of AA in the spectrum. But more commonly, spectra consist of superimposed mutations from multiple processes. So this is a lung cancer spectrum, and it's composed of a little bit of SBS1, which are these red peaks, a little bit of SBS2, which are these red peaks, which are different than these red peaks, a lot of SBS4, which we know to be the signature of tobacco exposure in the lungs, and then SBS5, which is a relatively flat signature found in many cancers. We think it's something that occurs naturally in all our cells, as does SBS1, and then SBS13, which actually usually occurs, co-occurs with SBS2. So this spectrum is then a linear combination of mutations from each of these 5 signatures. And there's the reconstructed spectrum. You see it looks quite, so this is basically composed by a coefficient for each of these signatures that then gives you, when added together, a spectrum. Okay, a reconstructed spectrum. Okay, and we often use cosine similarity as a measure of similarity, so this is quite similar. So data mining can tease apart signatures in large sets of spectra. There are approaches based on non-negative factorization, and there are approaches based on probabilistic topic models, which we won't discuss. I presume these slides could be made available after the talk, so people can look up this if they need to. This is a kind of unsupervised machine learning. As we've experimented with different methods, we find that they all seem to face similar challenges. And it's also important to note, and I'll emphasize this several times, signature discovery or extraction is not a purely algorithmic process. You really have to think about it and consider the results of the algorithm in the light of all available evidence. And part of the reason is that the granularity of extracted signatures and the type of mutations considered depend on the larger questions that you as a biologist might be considering. And so I showed one example of the kinds of mutational features, but one could also generalize this to small insertions and deletions. One could look at doublet-based substitutions, and we could look at other characteristics of mutations as well and consider them as part of a different feature set for signatures. So again, many techniques for extraction are based on non-negative matrix factorization, NMF. We're given a matrix V of observed mutations of spectra. In this case, it represents sort of the work that we did recently. And this was about 23,000 tumors and 96 mutation types. So each column is a tumor. Each row is a type of mutation like CAG to CTG. And we want to find a factorization that approximates the matrix V as the matrix product of signatures. So we have a bunch of signatures of 96 mutation types, and exposures or coefficients of each of those signatures to each of those signatures in each tumor. So we want to approximate that. We want to do an approximate factorization. And here, each cell contains the level of exposure of one tumor to one signature. And our objective function is going to be how close is this estimation of twiddle V, which is the product of W times H, to the actual observed mutations. One possible objective function would be the Frobenius norm of the difference between twiddle V and V, which is a kind of a generalized Euclidean distance. So some general observations on NMF. It's a collection of algorithms and techniques. The best approximate factorization depends on the objective function, which should be obvious, but often is not thought of. As for many unsupervised learning approaches, determining the number of signatures to discover is challenging. I will emphasize this. So this turns out to be quite an important challenge in practice. Okay. And it's also important to avoid overfitting, which is related to the previous bullet. So techniques often do multiple factorizations on bootstrap data to try to avoid overfitting in the machine learning. So I will just not go through this slide. It'll be there for people to refer to later. So this particular software, SigProfilerExtractor, has one approach to selecting the number of signatures. So it looks at the reconstruction error, which is red, and then the stability of the signatures, which is based on clustering the signatures across all the iterations, right? So one NMF might come up with slightly different signatures than another NMF, and you have to combine them back together again by clustering. And if the clusters aren't very stable, you probably overfit. So it tries to find a balance between the accuracy here in the red and the stability in blue, right? So here you get much more accurate reconstruction, but the stability starts to go down. So it tries to find some place, but it's not very clear exactly where you should go in many cases. Okay. So looking at the time, I'm going to skip through a few slides because part of this talk is stuff that I had not done before. So I wasn't sure how long it was going to take. So I'm not going to skip ahead to some important unsolved problems in mutational signature analysis. So remember we talked about just now extraction and discovery, but that actually requires a lot of computation. And I think not that many groups are going to be involved in extraction and discovery. I think many more groups are going to be sequencing 100 tumors or something like that, and they're just going to want to know what known signatures are present in these tumors. And that's really not necessarily suitable for extraction. You can do extraction, and it's useful in the sense that you can see what you extracted and compare it to what you get by doing attribution only. But mostly you're going to be interested in just knowing what known signatures are present. It's hard to find new signatures in a few tumors unless they're blazingly obvious. So we're going to talk about that. So there's some general issues with attribution. It's not very well studied compared to extraction, actually. One thing is that with enough signatures, adding more signatures almost always improves the reconstruction. And that raises some issues of sparsity. So you get tiny exposures to many signatures. And so now with some 50-odd signatures known, you end up getting lots of signatures helping to improve the reconstruction if you think about this just from a purely mathematical perspective. And that then raises biological plausibility issues. So you see the signature of UV radiation in brain tumors, or you see the signature of tobacco exposure in the lungs, in leukemias, for example. So I actually just talked about those. So these are some of those examples of biological plausibility or implausibility. So here's an example of several lung cancers where I simply optimized the coefficients of all known signatures to minimize the reconstruction error, the Euclidean reconstruction error, and ended up with 45 out of those signatures assigned. That provided really good reconstructions, but very bad model of reality. So we have, again, these are lung cancers. So we have 3 ultraviolet signatures. We have the signature of aflatoxin exposure, which is a liver cancer toxin, for example. So we have implausible biology as the result of purely optimizing the mathematics without incorporating any prior information. So we can, of course, do better. We could impose some sparsity by just requiring a minimum number of mutations from each signature. And that gets us down to just 30 signatures assigned, but it's still a good reconstruction, but it's still a bad model of reality. And in fact, the signatures in black, the signatures in red are signatures that have never been observed in lung cancer, and their presence in one of these bar graphs is indicated by these black rectangles. Okay. So we could also then, of course, consider only mutations found in lung cancer. This is actually squamous lung cancer, which actually has a simpler set of mutations, simpler set of signatures than adenocarcinomas. And we do much better, but there's still 4 signatures that are attributed that are actually very rare in squamous cell lung cancer. So it's highly unlikely that all of these would have had these very rare signatures in lung cancer. They're not rare in general, but they're rare in lung cancer. So now we're going to take a quick detour before we come back to a possible solution to that. And I'll talk about the signature presence test. So if you're given no mutational signatures, what's the evidence that a signature of interest is present in a particular spectrum? So this again was motivated by our work with AA, and we wanted to know which of these liver cancers has the AA signature. So we developed something called the signature presence test. And as I said, this is motivated by our study of AA exposure in liver cancer. So here's a spectrum of a liver cancer. So is the AA signature present? So looking at it, it's hard to tell. There's some gray stuff here, which could be due to this, but it's hard to say, right? Because our brains can't think about that much data. So what we did is we develop a best reconstruction of the spectrum without using the AA signature, and we get the log likelihood of that reconstruction. So that is the likelihood of the reconstruction. I should say the likelihood of the reconstruction is the probability that negative binomial resampling from the reconstruction yields this spectrum, yields this spectrum, okay? Right? And then we reconstruct the spectrum with the AA signature, and we get another log likelihood here. And we do a standard test, which is called the likelihood ratio test. And at this point now we've added a statistical concept, and we can give you a p-value for the null hypothesis that AA signature is not present compared to the alternative hypothesis that AA signature is present, okay? So here we reject the null hypothesis that the reconstruction of the spectrum without AA is as good as the reconstruction with the AA signature. And to put it maybe a little bit more easily understood, we found out that we need the AA signature to plausibly explain the spectrum. Okay, and we can extend this signature present test to sets of signatures, and we can ask the question, is this set of signatures needed to plausibly account for this spectrum? This means we can find all subsets of signatures that can plausibly account for a spectrum, right? So we subtract out the ones that aren't needed, and that's the ones that are needed. And then we can search among the subsets of signatures that can plausibly explain the spectrum to find the best of those subsets. And an example of multiple plausible reconstructions, here's one squamous cell cancer from the set that I just showed you. It has these possible signatures, and it turns out that we can actually remove 60 subsets of signatures and still get plausible reconstruction. So here's some examples of the subsets that can be removed and still leave us with a plausible reconstruction. So that then gets us back to the attribution only problem. And again, when we considered only mutations found in lung cancer, we did much better than previously, but we still had 4 signatures that are very, very rare attributed to each of the lung cancers. So the notion of the best subset among the subsets that can explain the spectrum, well, we're going to use a maximum a posteriori probability to find the best attribution, that is the best subset of signatures. And so we're going to maximize the probability of the data, the spectrum, given the model, and the model is the attribution times the probability of the attributions, where again, P, the probability of the data given the model is the likelihood of the attribution, the probability of the spectrum given the attribution. And for the probability of the model, we're going to use the product of the probabilities that each signature is present or absent in the given cancer type. And when we do that, we actually get a very plausible result. So these signatures, which are found in less than 1% of this kind of lung cancer, are no longer attributed. And we get SBS40, which is found in about 5% of these lung cancers, attributed to 1. And probably if I were doing this, really, I would then delve more deeply and find out which particular characteristics of the spectrum made it desirable to include SBS40 in the attribution of this signatures to this lung cancer. Okay. So now I'll just wrap up here. In the overview, I explained the concept of mutational signatures, and I gave an example of mutational signatures in molecular epidemiology. Mutational signatures revealed the role of AA, aristolochic acid, in causing liver cancer and other cancers. And that was this signature here that showed us liver cancer in many parts of the world, especially in East Asia. We also looked at sort of 3 questions, computational questions, that we would like to answer with mutational signatures. So I talked, I think I misplaced that slide. Let me see, I have my other slide here. Hold on one sec. I think I may have hid these slides. Hold on one sec. Excuse me. I've got too many screens going. Okay, here, let's do it this way. I'll just show you the, okay. Okay, so to summarize, we looked at, let's see, is this still visible? The screen sharing still visible?
Yes.
Okay, good. So we looked at extraction and discovery of new signatures. We discussed approaches based on non-negative matrix factorization and mentioned, but did not discuss approaches based on probabilistic topic models. We discussed the challenges, especially deciding on the number of signatures and the pitfalls, and the fact that discovering signatures is not a purely algorithmic process. In terms of attribution of known signatures, we looked at issues of sparsity. We observed that often many attributions, different attributions, can plausibly reconstruct a given spectrum, which suggested that we need to formally and informally incorporate prior knowledge. And we tried to capture some of this prior knowledge using by evaluating the set of plausible attributions using maximum a posteriori probability estimates. And I talked about the signature presence test, where we use a likelihood ratio test to decide if a particular signature or set of signatures is plausibly required to reconstruct an observed spectrum. Okay, and so I will take a chance here, see if I can go back. Yeah, looks like I can. Oh, and here it was. Okay, maybe it was lagging. Okay, so I just want to flash this. It'll be on the slides for people to look at later. This was the AA liver cancer paper, and these are some general acknowledgements. The work on extracting signatures from 24,000 tumors was part of the International Cancer Genome and TCGA Pan-Cancer Analysis of Whole Genomes Consortium called PCOG, and I co-led the PCOG Mutational Signatures Analysis Working Group with Mike Stratton, and there were many contributors, including Ludmila Alexandrov, Jaegil Kim, Gad Getz, Nick Hardbala, and many others. And I'd also like to acknowledge many people in my lab working on this. So again, there's a conceptual hazard here, which was brought home to me over the years working with this working group. These analytical tools are not oracles. They do not algorithmically reveal a Platonic truth, right? Their analyses need to be assessed by humans in the light of all available evidence. Okay, that's really, really important if you're going to use some of these tools, because I've seen very nonsensical results presented in papers. So my favorite motto is all models are wrong, but some are useful. And I hope that you will find some of these models useful, and we definitely need to do more research. And with that, I'll say thank you and take questions.
Thank you very much, Dr. Rozen. So I already noticed that their first question appears in the chat session and or several questions. Could you read and answer these questions?
Yeah. Okay. Let's see. Where's the first question? Okay. We highly encourage participations. Is there any? So the first one is from Guo Jun Sheng.
Yeah, yes, I think so.
Yeah. Okay. Very nice talk. Is any major difference in mutational signatures between primary and metastatic tumors? The only time we have seen that is when the patient was treated with chemotherapy prior to the metastasis, in which case the metastasis will have the mutational signature of the chemotherapeutic. But we haven't seen this in other situations. Then we have a question from Shofi Andari. I'm curious if correlations among the signatures, hold on, I have a little thing in the way here. Correlations among the signatures would matter in the model, especially to assess twiddle the V tilde. That's something I haven't thought about, to be honest with you. I can generalize from your question, but I can't answer your question precisely. I don't know if correlations affect NMF in general. What I do know is, of course, is that if you don't take into consideration correlations, you're leaving information on the table that you're not using. That's an open question as far as I don't know the answer to that, the first part of your question. I noticed that some signatures might give a number of zero values. I have I think of something that's floating over. I noticed that some signatures might give a number of zero values. Is this why negative binomial approach is used to compute the likelihood? The basic reason is basically the negative binomial approach is used to model the biological sampling from the idealized signature process, right? And we could do a Poisson distribution on each peak, but it's probably, because in biology things tend to be over dispersed, we're basically using the negative binomial distribution as an over dispersed Poisson distribution. You could also try to use some kind of over dispersed version of a multinomial, but we just are using the negative binomial. So the real reason is that we think this is, being biology, that there's some degree of over dispersion, and we're trying to capture that with the negative binomial distribution. Let's see, there's then, thank you, you're welcome for everybody. We're back to the, wait, then the metastatic question came back again, but I already answered that. Oh, I think I went to the bottom. Okay, hold on. Okay, is there a correlation we could draw between mutational signatures and genetic signatures? So I'm not sure what the question, what do you mean by genetic signatures? I can think of two connections. One is we could look at which drivers are being mutated by mutational signatures. So that's one kind of connection to genetics, right? And the other thing we could look at is, are there any genetic changes that create mutational signatures? And there are. So we know that mutations, either inherited or somatic mutations in the proofreading domain of one of our DNA polymerases, has a very distinctive mutational signature. Was somebody ever trying to clarify the question? Yeah. And of course, the well-known case of mismatch repair deficiency in microsatellite unstable cancers, MSI cancers. And MSI cancers have obviously a distinctive mutational signature in the insertions and deletions, and they also have a distinctive signature in the single base substitutions.
Thanks.
Yeah.
May I add to one more question? Maybe my question is not so correct, but I am also interested in how each mitogen or pathogen can be characterized with each signature. So my question is, if we, for example, cluster the signatures based on their mutual similarities,
yes,
can we guess that this cluster of signatures are related to some related to chemical natures or some related to mechanisms of mutations or that kind of thing?
To some extent we can. So it turns out there are a lot of exogenous mutagens that form adducts on guanines, and they all tend to form mutational signatures that are heavily dominated by the blue stuff at the far right hand of the spectra. So those would include aflatoxins and nitrosamines, but in general, it's a hard problem, and partly because we don't have that much data, right, on different mutagens. Yeah.
Another question came out.
Okay. Okay. So a clarification. So Prash, in lieu of compounds.
Yeah, Professor Rozen, I apologize now for unmuting.
That's fine, please.
I seek a small permission from Professor Kenta-san. Yeah, my question was in lieu of this, Professor Rozen,
Yes.
that from the AAs or from the the wonderful analysis that you have analyzed, taking the spectral data, we could definitely look into the so-called the genetic signatures that these harbor, but at the same time from the exomes, from the mutational signatures that we could derive from, there must be a common correlation that could be drawn. Say from the same spectral data, could there be any kind of mutational signatures that we could that we could draw from, that we could obtain from? That was my point. Yeah. Thank you.
So draw from genetic changes in human cells?
Yeah, exactly. Yeah,
right.
Please.
Yeah, so there are signatures from genetic changes in human cells or mouse cells or whatever, right? And I can kind of list these. So there, I talked about the mismatch repair deficiency signatures found in MSI, microsatellite unstable cancers, right? And they're the polymerase epsilon proofreading domain mutations, which cause a very distinctive mutational signature. There's a MET-YH deficiency signature. We also found that one particular hotspot mutation in topoisomerase 2A causes a very distinctive insertion and deletion signature in human cancers. So there are genetic causes for some of these signatures.
Right. Thank you so much. Thank you.
Yeah.
Thanks, Dr. Rozen. It is about time. So let's thank the speaker again. And sorry, you can't hear the.